Hello. Questions are in. 1, 2 from the year 7 booklet. I would happily do that now. Um, I should also just say well, I'll say it in a minute once more people arrive. Um, Questions 1 and 2 from the year 7 booklet, which is here, I believe. So everybody in Key Stage 3 did some stuff on the digestive system and also the circulatory system, which is the heart and stuff. A lot of this, especially if you're in year 7, will be challenging, but that's okay because it will get easier. Um, The first question on here says foods are digested before they are absorbed into the blood. And the diagram shows organs in the human digestive system. Just on this, I'll just do it all very quickly. But A is pointing at the mouth, right? But it could also be pointing at salivary glands, which are also in your mouth and are part of your digestive system. B is pointing at your liver. C is pointing at your stomach. And D is pointing at your small intestines, which should answer a question. Which organ is the stomach? C. And what type of enzyme is produced in the stomach? Okay, I'm going to do a very quick summary here of things. There are three enzymes: amylase, protease, lipase. They have different functions, but we'll come to those in a minute. In terms of where they are made, there are three places for amylase. Those three places are salivary glands, small intestine, and pancreas. I appreciate that's just letters. Protease is produced in the stomach, the pancreas, and the small intestine. And then lipase is produced in the pancreas and small intestine, which means when it says which type of enzyme is produced in the stomach, if you know this, then you know that protease is the one that's produced in the stomach. Which term describes the pH in the stomach and give reason why this is the pH? Well, the stomach um, produces a substance called hydrochloric acid. The reason it produces that mainly is to protect you from bacteria on any food that you may have eaten. And so the answer to this question is acidic. And the reason for it is because, well, it produces hydrochloric acid. Okay. And which organ produces bile? You need to remember this. The thing about bile, the least to try and remember, there is a few things, but bile is made in the liver, but annoyingly, it is stored somewhere else. It is stored in the gallbladder. So when the question says where is it produced or made, the answer is the liver. Booklet 3, question 2. Yes, I shall do that in just a moment. How does bile help in the digestion of foods? The answer to this question is it increases the surface area of fats. What it basically does is it takes a fat molecule and it sort of cuts it up into smaller pieces, which means that when we try and digest this, it is much quicker to digest this than it is to try and digest this big lump. And a student tested different foods for the presence of protein, starch, and sugar. These are all just things that you will gradually learn, which is that when you test for these foods, there are different solutions that you use to do the test. For protein, it is this stuff here. For starch, it is this one here. And for sugar, it is that one there. A safety precaution you should take when using Benedict solution. I'm going to say wear gloves or prevent it from getting on your skin, but I'm just going to check what the mark scheme offers for that. Um, Wear goggles, wash spills. Yeah, that goggles, let's just say. And I'll just get through these two questions and I'll move to booklet 3. The table shows the results for one food sample. Which of the tests show positive results? Okay, so this test, okay, this solution, Benedict solution, is blue to begin with. And if there is sugar present, then it changes to red. So that's what's called a positive result, as in it changes color. So the Benedict's has changed color, so that's one. Biuret is a solution that begins blue, and when we're testing for protein, it changes color to like a pink or a purple. So if it stays blue, it means it's not a positive test. Iodine is starts off orange, and when we test for starch, it changes to blue, black. Oh, let's talk black. So if it changes black, it is a positive result. So the two positive results are the Benedicts and the iodine only. And starch molecules are not absorbed into the blood from the digestive system. Why? Well, starch is too big. And that's why we have to digest big molecules into smaller ones. And starch is an example of a big one. Right, question number two as well. Human breathing system. Number one is called the trachea. And then alveoli, I think. Oh no, it's just lung. Right, right, lung. When we breathe out, the mixture of gases which leave the air sacs contains more. So breathing out, what do we breathe out more of? Carbon dioxide. You can write the name in or just put that in. And less oxygen, basically. So we still breathe out a bit of oxygen because it sort of mixes in the same place, but there's much more carbon dioxide out. Oxygen, I should say. And that's it, isn't it? First two questions. Okay, right. I'll move on to booklet three. There won't be many questions today, I don't think. It wasn't a terribly hard, these homeworks, I think. Booklet three, question two, and then I'll do 3A part two as well. So question two on booklet three. Here is the digestive system. Which organ produces acid? Acid is produced by our stomach. So we need to just point at the one which is the stomach, and C is the stomach. Which organ produces bile? Bile is produced by your liver, and the liver on that diagram is this thing here. So B. And where are foods absorbed into the blood? So eventually, when you eat food, it eventually ends up in your small intestine, from where it is absorbed into your blood from there. Once it, because by then it's small enough. Um, and so E, is that it? Yeah, small intestine, E. Um, and food molecules such as proteins cannot be absorbed. Why? The three large molecules that we eat, as we saw in the question that I did before, are starch, protein, and fat. These are large and they have to be broken down into smaller ones. So when the question says why they can't be absorbed until they're digested, it's because they're too big. Proteins are too big. Um, I'll just check I'm still doing the same question here. Um, okay. Yeah, that's it. It's too big. And then the next question says, yes, I'll go through all these questions. I'm just doing in order that they're being asked. What is the optimum pH for enzyme Y? Optimum means the pH at which it works best. So the highest point of this graph is where it works best. And so that is at pH. I don't know what that is actually. And then Y. So it's not 7. I go for 7.6 roughly. 7.6, 7.5. Somewhere around there. Should be fine. Um, and then where in the digestive system might the two protease enzymes be produced? Protease is produced in three places. They are the stomach, the pancreas, and the small intestine. So out of these, oh wait, oh wait a minute. Right, it's a bit tricky this one, but these two enzymes, one of them is produced where the pH is very low, and the other one is produced where it's normal. So the low place will be the stomach. Remember, the stomach has a low pH, it's acidic. So I think it's that one. This shows a model of an enzyme molecule. Which substrate fits the enzyme molecule? You need to find that. This is like a jigsaw thing, isn't it? Isn't it this one? Because that piece will fit into there, and then these two pieces will fit into there. I think that's it. Um, it's the only one that makes sense to me. The enzyme or substrate is used for a model of enzyme action. What is the name? It's called a lock and key. So this idea that you've got to get the right shape to fit into another one, that's what it is. Okay, that was question. I should need to open this up actually. Just give me one moment. Okay. And then the next question, 3A part two. Oh, I didn't finish this off today. Explain why pH affects enzyme activity. So you want to say that when the pH is low, um, can denature an enzyme. Denature means that that shape that we keep looking at in the previous thing, the shape changes. And so if the shape changes, it no longer fits the food type or the substrate. The substrate is just a fancy name for, in this case, the food type that fits into that thing. I'm just going to 3A part. I'll do that one first, and I'm just going to move on. 3A, 2. What is the main tissue in the heart wall? It's called muscle. It is a muscle. The heart is just a muscle. Okay, right. I'm just going to revert back quickly to oh, I did question one. Right, I'll show you the answers quickly to question one in booklet one. So you've just arrived a little bit later. But in question one in booklet one, we said that the stomach is organ C. The enzyme that's produced in the stomach is called protease. The reason the stomach is this pH is it's acidic because it produces a hydrochloric acid. And the organ that produces bile is called the liver. The way bile helps in the digestion of foods is it does this, which increases the surface area. You might not have learned that yet. And then these food tests, protein, we test for using something called Biuret reagent. Starch, we test for using iodine, and sugar, we test for using Benedict's. You will gradually just remember all of this stuff. And the safety precaution is goggles. Um, then again, this doesn't help, but the answer is Benedict's and iodine for this one. I did explain it a little while ago if you want to watch it back. And then the reason anything is not absorbed into the blood is because it's too big. We have to make it smaller, essentially. Right, and we'll go back to this question. One, part one, I'm just going to do that. And then I'll move to question five from here. One, I. Right. Right. So the question is, how are these things called villi? These villi are found on the lining of your small intestine. So millions of these little things. And the good things about these is that they have their job is to take your food, which is in here, once it's small enough, and absorb it and put it into the blood. And they can do that really quickly because they've got very thin walls, which results in a reduced distance for diffusion. Diffusion happens much quicker across a thin wall. And so that efficiently absorbs this sugar. And the other thing that villi have that's really good, by having so many of them, they have a large surface area. And what a large surface area does is it results in faster diffusion. Similar to the one above, really. But it's just that lots of diffusion can happen. Or it maximizes diffusion. I think I might have change that to maximizes diffusion. And that should be 4 marks because you have to say one and then explain it. Question five. No, not question five. Six. 6C even. Give two ways in which the composition of blood changes. It flows through the vessels labeled X on the diagram. Okay, so let me just explain what's going on here briefly. I'm gonna do the whole thing just very quickly for everybody. That'll be helpful. These are your lungs. What happens at your lungs? You breathe in oxygen. That oxygen is then taken to your heart, if you just follow my arrow. It is then pumped from the heart all around your body, everywhere. So it goes to your head and whatever this says, head and upper body. Some of it goes to your liver and your gut. Some of it goes to the rest of your body. Eventually, it comes back, right? What happens here in all of these places is the oxygen goes in that we've just brought from the lungs, and it uses the oxygen, or all your cells use oxygen for respiration, which is to get energy. A waste product of respiration is carbon dioxide. And so that carbon dioxide is kind of being produced here, and we want to get rid of it. So this blood now brings all of that carbon dioxide back, firstly to the heart, and then the heart sends it to the lungs, and we get rid of it from there. So when it says, explain, oh, I can't remember what the question is. Give two ways the composition of the blood changes as it flows through X. Well, as it flows through X, the amount of carbon dioxide in the blood goes down because we get rid of it, and the amount of oxygen in the blood goes up because we take more in. So the answer is, I think, just less carbon dioxide and more oxygen. Okay. 5E in that one. I thought I did that earlier. Um, how does bile help the digestion of food? It is this answer here. The increases the surface area of the fat. It's a few repetition of these things. Six, I think I just did, didn't I? Yeah, not all of it. Well, okay, fine, I'll do all of six. Name the type of blood vessels labeled A, B, and C on the diagram. Okay, let me get rid of all of this. So, the way to remember this is that blood vessels, there's three options. There's arteries, there's veins, and there's these things called capillaries. Okay, so anything where the blood is going away from the heart is an artery. The way to remember that is it starts with an A. Anything where the blood is going towards the heart is called a vein. It's going into the heart, and veins have the word in them. Trying to remember that. And a capillary is the bit where anything changes. So oxygen goes in or carbon dioxide goes out. So all of these things will be capillaries. So B is pointing at a capillary because that's where oxygen goes into your cells and carbon dioxide comes out of your cells. So B should be capillary. A is, if we think about the direction of A, where is it coming from? It's going away from the heart, right? Away from the heart, away from the heart. So there would be arteries. And C, what's happening here? It's going back into the heart from here. So that is a vein. The job of the circulatory system, I'm just going to say, it is to um, um, carry oxygen around the body and remove waste products. I think that should be sufficient for that answer. And I did see. Question five, booklet one. Booklet three, question 6C. Did I just do that? I just did that. Is that all of them? Amazing, efficient. Um, I will just open up something else that I think is important for everybody. So we did two topics there, digestion and a little bit about the heart and stuff. Um, the one important thing to try and remember is everything you see in this table, because if you can remember everything in this table, then it kind of answers a lot of the questions that you get on the digestive system. We saw a lot of them already, like where the enzyme was made. Um, but each of these enzymes, three of them, have a job, which is to break down a large type of food, starch for amylase, protein for protease, lipids, which are fat for lipase. And then these big things get broken down into smaller things. The smaller things are named there, sugars, proteins, amino acids, and fatty acids and glycerol. And then where those enzymes are made is here. So if you've learned this table, then actually a lot of those questions become much more straightforward. Um, and then there was a question on bile. And then the only other thing that I should just do while anybody asks me any other questions is the food tests. Okay, the sooner you guys can learn these, your life will be so much easier. So there is a food test involving sugar. There's a food test involving starch, food test involving protein, and a food test involving fat. And by involving, I mean if I wanted to find out whether a biscuit, for example, contains some sugar, I can do a little test to find out. Similarly, if I want to find out if it contains starch or protein or fat, what it involves is getting a test tube, crushing up a little bit of your biscuit or whatever it might be, and putting it in some water. That becomes your sample that you're going to test. And into that sample, depending on which of these four you're testing for, you add a different solution. The solution you add for sugar is called Benedict's solution. For starch, it's called iodine. For protein, it's called biuret. And for fat, there's two, but I'm going to go with the one that's called ethanol. So we add these in, and all of these have a color to begin with. So Benedict solution is blue to begin with, iodine is orange to begin with, biuret is blue to begin with, and ethanol is colorless to begin with. It's clear. Clear, let's put clear. Right. So it's that to begin with. If the food sample that I made contains sugar, then the Benedict solution will change from blue to another color. It usually is red, but it can be a range of colors. If the food sample contains starch, then when I add the iodine solution, the iodine will change from orange to black. If it doesn't contain starch, it won't change color, it'll stay orange. For protein, Biuret starts blue and it changes to pink or purple if we've got protein in this biscuit, or whatever it might be. And fat, if we're testing for fat, we add ethanol and it changes from clear to like a cloudy white color. These, if you can remember these, it does make life a lot easier. And then I'll do one more thing, which I think is important for the heart and stuff. So here are your lungs. If you can recreate what I'm about to do, it is very helpful. Here are your lungs. Here is the heart. Okay. And here is the rest of your body where all of your blood goes to, right? So everywhere. That can mean your brain or your legs or your hair or whatever it might be. Okay. Right. So that's just everywhere. You breathe in oxygen into your lungs. You're doing it right now. The oxygen goes into your blood. The blood then travels to this side of the heart first. Make that red, which is actually the left side of the heart, confusingly, because it's the person's left, not as you see it now. It's then pumped out of there all around the body. Okay, so that oxygen that we breathed in first goes to the heart and then it's sent everywhere. The oxygen goes into all of your cells, and all of your cells give back some carbon dioxide because respiration, remember, is glucose plus oxygen gives carbon dioxide plus water. So we're using the oxygen and some glucose to make energy. Carbon dioxide is a waste product that we want to get rid of. So the cells go, right, can you get rid of this carbon dioxide? Your blood then takes the carbon dioxide back, but to the other side of the heart, so it doesn't mix with the other one. And then the heart pumps it back to the lungs where we then breathe out carbon dioxide and the whole thing starts again. That little simplified structure helps you understand the process. And then there's more detail within that, like the names of the blood vessels. There's like things called pulmonary arteries and pulmonary veins and aortas. But if you step back from it and say, well, this is the loose structure here, then the rest of it hopefully becomes easier. Are there any other questions from any of the homework booklets? Nobody from booklet two. I don't know if this was very similar to. Yeah, I think it was actually. I think that was basically the same as three. Good. I'll just see what else there was in this. Let me know what the answer to this question is. I'm only asking because it's got nothing to do with the specific topic that we did today. So there are two types of microscopes. There is a light microscope, which is the one you most commonly used in school. And there is something called an electron microscope. The electron microscopes are more expensive because they're more powerful, but we don't want to say more powerful. There are two possible things you can say about why they're better. You can say that they have a higher magnification, which means they can zoom in further, or that they have a higher resolution, which means that you get a sharper, clearer image when you do this. Oh, I should also add, you guys will be doing a test, just so you know. You get the advantage of knowing. But I did send an email about it a week or so ago, but not this weekend if you attend on a weekend, but the weekend after, just so you're aware, there will be a test. The topics will be things you've done recently, like photosynthesis and maybe disease and forces, maybe in physics. I can't remember any forces. But yeah, I'll send a list around, but just so you know, that weekend, roughly, anything from beyond the 15th onwards, there are tests for that week, just so you know. Right, given that there are no other further questions at the moment, I'm going to come back shortly when I do the year 10, 11 stuff. But if you do have any other questions, I'll keep checking and I'll pop back in and check. Hello, whoever has a question, you can let me know. I am still here if you want to ask me a specific question from the booklet. Tell me which booklet number. Okay. Question number 1E. I think I did that earlier. 1E, booklet 2. Okay. Yeah, that's a little bit harder to do. But um, well, it says calculate the image length, which is there, of something that's viewed at a magnification of 20. So I'm going to change that, put 20 there. The real object, it says, is 0.8. Well, that's real, 0.8. So it's image divided by 0.8. So if you saw a question like this and you had to figure out what the image was, you would do 20 times 0.8. Um, that's all you would do, 20 times 0.8. And you work out what that is. Um, 16, I think. And it's just 16. That's it. It's about putting the numbers into the formula um, that you have there. And then you would have to do a bit of rearranging. It's very common in physics where you have to use an equation that they give you to put some numbers in. Anything else? No problem. Okay. Hello. Hmm. Does anybody have any questions on the other homeworks that we set this week? So year 10, I think, did a recap of cells and respiration, aerobic, anaerobic. I think all the beginnings really of biology. And then year 11, I just got a mixture of things now, obviously. If you're here, I might end up just going through some of these because it'll be applicable to everybody as we go. Right, if you do have any questions from the year 10 homework at all, then just feel free to um, tell me. Otherwise, I'm going to start going through some of these because this will be applicable to everybody. Because there's some quite complicated questions in here that I think would be interesting to for me to do. Um, the table shows the actually, yeah. The table shows the concentration of three mineral ions in the roots of a plant and in the water in the surrounding soil. The plant roots could not have absorbed these mineral ions by diffusion. So the reason is because over here we have a low concentration of things, and over here we have a high concentration, and we're trying to take these up from the soil. Diffusion is not low to high concentration. Diffusion is high to low concentration. And to take these things up the soil, we're going to have to use, well, always explain that diffusion is high to low, and in the soil there is a lower concentration. That's what I would say. Name that process. Well, that process is called active transport when we absorb things from a low to a high concentration. That is what active transport is. How do the following features of plant roots help the plant absorb ions from the soil? Well, by having thousands of these root hairs, whenever you have a lot of something in biology, you kind of want to use the phrase large surface area. Um, yeah, that's it. No more questions. That should arrive hopefully by Thursday, Friday. It's just they wouldn't have gone on Monday for homework. It does happen. I think you should have received an email with it electronically though. But if not, then tell me. But you should have done because I think I submitted it there and then um, after the lesson. But it is this one. But yeah, this does happen on a Sunday. The problem is the Sundays, isn't it? Because it doesn't go until a couple of days later. But also it's a bank holiday, so it will might be delayed again, unfortunately. But it's okay because you'll get it. Um, this is an important theme, right? So you should know that active transport is a process that requires energy. This is what we must do, which means that wherever active transport takes place, um, because you need energy, where does the energy come from? It comes from a process called respiration, which happens inside these mitochondria. And so this root hair cell that sometimes has to do active transport, it needs energy from aerobic respiration. So we want to explain that respiration releases energy, or the energy that it needs for active transport. There you go. Many of, oh, right, hang on. What's this? Sorry. How do the following features help them absorb mineral ions from the soil? Many of the cells in the root store starch. Well, in order for respiration to happen, we need two things. We need some glucose and some oxygen. So this store of starch is your basically your source for that to be used for respiration because it can be converted back to glucose. We have another question. Um, Yeah, so again, the reason you wouldn't have got any note partly because it wouldn't have been posted until probably Tuesday yesterday, just because of the bank holiday weekend. It wouldn't Nothing would have gone until then. So that's fine. This happens if you attend online over the weekend. It will be a delay, but it's okay. Just if you have any questions on it, then if you can't hand it in on the next lesson, just ask about it next week or watch the video in case somebody asks me some of those questions from those. Um, but what I'll do is I'm going to go through these questions and that'll probably be it for today. Um, but if anybody has any other stuff, especially if you are in year 11 and you have time or the clock ticking, so to speak, then please do use these to ask me anything. Um, Draw one line from each disease to the pathogen that caused the disease. I should also remind you, in case you've just joined, that everybody will have a test in a couple of weeks' time. Um, so yeah, the week beginning the 15th. Um, The pathogen that causes gonorrhea, which is an STD, is a bacteria. Malaria is caused by this thing called a protist, and measles are caused by a virus. Um, Malaria, as you may know, is transmitted by mosquitoes. They bite one person and then they bite another. Male mosquitoes can be sterilized, so they are infertile. The spread is reduced by releasing sterile mosquitoes into the environment. Explain how that reduces the spread of malaria. Well, I presume if they're infertile, it means that they don't reproduce. And so it reduces reproduction of mosquitoes. And if you reduce reproduction, what does that mean? That there's fewer mosquitoes. And if there's fewer mosquitoes, they don't bite as many people, which means that they don't pass on the pathogen to others. Pathogens also cause disease in plants. The figure below shows a rose black spot fungal spore and a tobacco mosaic spore. Okay. The piece of equipment used to view this is probably a microscope, but I think you need to be specific about the type of microscope. It would need to be an electron microscope because of the fact that these things, like viruses, are very, very small. You would not be able to see them with an ordinary light microscope. How many times longer is one than the other? Use the figure above. Ugh. Okay. Right. I have got something that is 16 micrometers long, and I've got something else that is this many meters long. Right. I cannot compare these two until I change this to the same as that unit. So I'll show you how I would do this. This number is meters, as it tells you in the question. Okay. To turn meters into centimeters, I times by 100 because there are 100 centimeters in a meter. To turn centimeters into millimeters, I times by 10 because there are 10 millimeters in a centimeter. And then to turn millimeters into micrometers, there are 1,000 micrometers in a millimeter, which means that this number in micrometers is 0.25 micrometers. Um, and then to figure out how many times bigger this is than this, you just divide them, right? So 16 divided by that will tell you that it's 64 times bigger. I assume that's the answer. It is. Explain why plants. That's a great question. This tobacco mosaic virus is like when you have a leaf and it has like these black patches on it or dark patches on it, brownish patches. In other words, they're not green, more importantly. Explain why plants infected with this grow slowly. Well, what this disease does is it causes a lack of chlorophyll because if a leaf is not as green as it should be, then there is a lack of chlorophyll in that leaf. What does chlorophyll do? It absorbs light. So if there's less chlorophyll, there would be less light absorbed. Why is it absorbing light? It is absorbing light to do a process called photosynthesis. So if it's absorbing less light, there would be less photosynthesis taking place. What does photosynthesis do? It produces glucose for the plant to help it to grow. And so less glucose would be produced. And that is the reason why it grows slowly. So you got to go through that chain of events, um, basically. Um, you could say the less glucose, you probably want to say less glucose for respiration. So there's less energy for growth, that kind of thing. Or there's other things you can say as well. I'm not going to read all of that. Okay, let's do one of these. So draw a Punnett square. That's one of these things to show the offspring from a male homozygous dominant. If it's someone's homozygous dominant, dominant is a capital letter. Homozygous means they're both the same, so they'd be both capital letters. Doesn't matter what letter to use, but I would just use H's. And a female homozygous recessive. If you're recessive, you've got the small letter, and if you're homozygous, they're both the same. So two little H's. And then you just, it's like a multiplication grid. So that would be, that's the combination you have here. That's the one there. That's the one there. And that's the one there. And that would be it. State the probability the offspring will have Huntington's disease. Well, the idea of this, for those of you that haven't done it, is that if you have the capital letter, that overrides the small letter. So the capital letter is basically makes you have this disease. So if all four of these possibilities have the disease, basically, so the answer would be 100% chance of the offspring having this disease in this situation. Okay, fine. Just in the year 10 homework, let's have a look at what was exciting about it. Um, this mentioned a light receptor cell. You had to name the structures, things like cell membrane, cytoplasm, nucleus. Um, then same sort of stuff again, labeling some cells. Movement. This cell is respiring aerobically. If it's respiring aerobically, what's the movement of oxygen? The oxygen has to get into the cell from a high to a low concentration, which to me looks like A. Um, and carbon dioxide moves out from a high to a low concentration, so that looks like B to me. That'll be that. This process is called diffusion, which represents the active uptake. Active uptake is like active transport, so low to high into the cell. That looks like it to me. Purpose of respiration is to release energy. Do try and say the word release when you get these questions, not just make energy or something like that. Balance this equation. Whenever you're balancing equation for respiration or photosynthesis, you put sixes on the smaller pieces. That's the way I think of it. And that substance is called glucose, the one with all the big one. Um, how are villi adapted for absorbing oxygen? You can say things like large surface area because there's so many of them. You can say thin walls. You can say even a good blood supply. It doesn't ask you to explain it, so we won't explain it. The gas which is excreted means removed from your body. So through the alveoli or your lungs, what gas are you excreting? Carbon dioxide. Name of the process of respiration when oxygen is not available. Read the question carefully. It's called anaerobic respiration. And describe that process. Well, the equation I think you should write down in the answer to this question, which is glucose gives lactic acid. Um, and you can mention that that happens generally during intense or vigorous exercise when you have a lack of oxygen. You might also want to mention that afterwards, you have an oxygen debt, which means that you carry on breathing heavily afterwards. Um, yeah, that's fine. I'm just going to go back to the year 11 question because there's a question on it. 4A. Did I not do that? I thought I did that. No, that was an earlier 4A. There's two different 4As. Oh, okay. Um, right. Low light intensity is one factor that limits the yield of a crop. In Britain, many tomato growers use artificial light to increase the yield of tomato crops. The table shows the amount of natural daylight and artificial lamplight received by a tomato crop in a greenhouse. Oh, wow. Right, lots of information. The question says, describe the pattern for the amount of light energy received from natural daylight by a tomato plant during the day. Natural daylight, so that's here, during the day. Um, describe the pattern. Well, the pattern to me suggests that it receives low amounts of light energy in the sort of winter months, like January to March and October to or November, December, January sort of thing. Low amounts during the winter months or when the days are shorter, because the days are also shorter. You see the number of hours. So shorter days, less light energy. Whereas in the summer, like in June, July, August, when the days are longer, so you have a longer days here, um, it receives more light energy. You also just probably want to put in some sort of numerical thing. So, for example, um, you want to say it's like compared to the winter, it's like, I mean, it's like more than 10 times higher, isn't it? So you see that number there. In March, it's like 848, whereas in July, it's like, sorry, not 10 times that. Yeah, so about from here, winter 183, it's like 10 times that. So 10, 11 times. That's some sort of numerical thing to show that it's a certain number of times more um, light there. That's it. So low in the winter, high in the summer, and then a certain number of times more than that. Is that all right before, eh? Um, This is a good question. I just want to do this. It's such a common question, this kind of thing. In an investigation, four groups of athletes were studied. The maximum rate of oxygen consumption was measured. So that's how much oxygen they can take in, basically. And we've got their time in a race. What is the relationship? Well, it looks like the bigger the maximum rate of oxygen consumption, the quicker your race time. You see that? So that's as that's bigger, that's a shorter time. In other words, you're probably able to run faster if you can get more oxygen into your body. Why? Well, if we can get more oxygen, so I'm going to simplify this answer. If you can get more oxygen into your body, you can respire more. And if you can respire more, you can release more energy. And if you can release more energy, you can probably run faster and therefore reduce your time in this race. It's a very common sort of question relating oxygen to energy by mentioning respiration in some way there. Not really the breathing question. The chemical process that releases energy is respiration. The substances produced, make sure you read the question carefully, carbon dioxide and water. Why does this have to take place more rapidly during exercise? Same answer, really. During exercise, you need more energy because your muscles are working harder. You would therefore need to respire more quickly to release more energy. See, if I was looking at this question, it says calculate the heart output for this person during exercise. So you need to figure out what this number is that goes in this box. Well, essentially what you should do is use this to help you and go, well, how might they have worked out this 5,400? Well, just check if it's 72 times 75. It is, which means that the number that goes in this box surely is 165 times 120. In other words, during exercise, because your heart is beating faster and harder, it's getting more blood out more quickly. And that's the same answer. We have the same question about three times there. During exercise, why is it more helpful to have more oxygen, more respiration, more energy released? That's it. There is a heart. Come out of nowhere. Which blood vessel carries deoxygenated blood? I did this earlier with the other students. I'll just tell you now that you should know this much. If these are your lungs and this is the rest of your body down here, in terms of the route that everything takes, is the following. You breathe in oxygen to your lungs. The blood flows into this side of the heart and then is pumped out all around the body, where all that oxygen that you breathed in goes into your cells for respiration. Those cells then give out carbon dioxide because that's a waste product from respiration, and your blood brings that carbon dioxide back to the other side of the heart and it's pumped back to your lungs, where you then breathe out the CO2 and then you breathe in more oxygen, and the whole thing starts again. It's a very simplified circulatory system. You should know this much. Oh, sorry. So the answer to the question, which blood vessel carries deoxygenated blood? This side here is deoxygenated. Which one carries it from the heart to the lungs? Well, that's here, isn't it? B. I can't remember the answer to this question. Is it right, Adrian? Is it a pacemaker? I'll have to check. Where does the pacemaker go? Right atrium. Well, the correct answer. It's just something to try and remember. Oh, this is good. Right. So in certainly in AQA, for those who are doing AQA at least, there is two drugs that you need to know which plant they come from. Those two are these two, digitalis and aspirin. Aspirin comes from a plant called willow, and digitalis comes from a plant called foxgloves, which, if you know that, this question becomes straightforward. There. And then there's loads of questions here about these, which you could have a look at. Usually, any question involving electron microscopes, you need to say higher magnification and higher resolution. They're kind of the two key things to say about them. Okay. Well, this is to do with stents and statins in terms of the question. Um, this artery has got this fatty material building up, and they're saying that as a result of that, the heart muscle starts to carry out anaerobic respiration. Well, why does anaerobic respiration happen? It's because of a lack of oxygen. Why is there a lack of oxygen? It's because we have narrowed this artery due to the fatty material, which means less blood flows through. And since less blood flows through, less oxygen reaches your heart. And if less oxygen reaches your heart, it starts to respire anaerobically because it still needs energy to pump. So, but it's not being provided with the oxygen that eventually can lead to a heart attack. Um, okay, well, that's all of that. Right. I'm just going to do one more thing, which was somebody sent some maths questions last week, and I'm going to try and just do one of them, given there's no other questions. But if there are, then please tell me. We have a bit of time. As a reminder, again, from the end of this month, for those of you in year 11, and year 10, if you really want to join, you're welcome to. I will be doing one of these, something like this every day. It's great fun. 11 part 3 at the back, yes. Oh, wow. Okay. Oh, where's that come from? Okay, let me just read this. Um, where's the food web? There's something missing here. Um, okay, I think there's something missing in that question. A parasite is essentially an organism that lives on a host organism. I think it's slightly example specific, but it's to do with what a protist essentially is. It is a type of parasite. The mosquitoes essentially feed off a host. So it is an organism that lives on a host organism or feeds off a living organism. Um, and it can cause harm to it, mainly because it's taking its food in some way. Um, oh, it's in, sorry, I'll just finish this one off. Oh yeah, that's weird, isn't it? Okay, fine. This food web's there. Right, okay, fine. And then can we do that? The leaves of stevia taste very sweet. People know how to grow stevia as a crop. You add, why do they do this? Oh, I see. Right, so random question, but if you are trying to grow more of this plant, what you don't want is for this plant to be eaten by these things here, because they will feed off it, right? That's what a food web shows. And so if you can add more of these to your field, those things will eat these things. Therefore, those things will not eat the stevia, which means you'll be able to get more stevia. Very unusual way of asking that question because, yeah, that's what it is. Right, question eight and the second question six here, 10. Let me have a look at that. Probably did that. Oh, no, I know. That's probably the one I missed out, isn't it? Um, oh, question eight. Okay, question eight and the second question six. Right, question eight was here. Deoxygenated blood away from the heart was B. Natural resting heart rate. There's something called a pacemaker. It's in your right atrium. I mentioned that digitalis is a drug that originates from something called foxgloves, a plant that you do need to know. And then this question. Um, question says, calculate stroke volume X in the table above. Okay, well, again, it's similar to that earlier question. You have to sort of figure out how these numbers have come about at the bottom. Now, I presume if you times these together, you'll get 8624, I presume. You do, which means 52 times something is 2,800, which means that that number there must be 53.8, and they've asked you to round it, don't they? Oh, sorry, I told you that anyway in the question. Never mind. Um, so it's 54, whatever that is. Yeah. Okay. Then, some people who take beta blockers get out of breath when they exercise. Explain why they can have this effect during exercise. Right. You should refer to the information. So, well, when you take these blockers, the cardiac output, which is the amount of blood being pumped out by your heart, significantly reduces compared to if you don't have those. You see that 5,540 if you at rest when you don't take them, and it's basically half almost. That similarly, when you're exercising, about half as much um, blood is being pumped out of the heart. It tells you in the question what that means. So you need to explain first that when you take these beta blockers at both rest and exercise, about half of the volume of blood is pumped out of your heart as it would if you were not taking them. You could explain why that is. It's because both your heart rate and your stroke volume are reduced, right? See that? Everything's reduced basically. Your heart is pumping out less blood. It's beating less fast, which means your cardiac output, which is the amount of blood being pumped out, is lower. So let's see, explain that first for the 6 mark question, that far less blood is being pumped out of your heart. Then explain the effect of that during exercise, because what is that blood doing? That blood is carrying oxygen. And a lot less oxygen, therefore, is getting around your body to your cells, for example, to your muscle cells, which means that you would respire less, um, which means you'd release less energy. And to compensate for that, what you try to do is increase your breathing, and therefore you start to get out of breath um, quickly because you literally have um, less oxygen getting around your body. So it's kind of explaining that table and then going into the next part. Right, I'm going to probably not do a lot more, but let me talk about a question, a maths question. Let's see what this is first. Okay, just one reasonably complicated math question, then we'll finish for today. Hmm. Right, you've all probably seen in maths a table of values of this sort. The reason this question is a little bit complicated is because it's a graph that you may not be used to seeing. Um, and I'll explain why. Whenever you have an equation which has an x on the bottom of a fraction, there's a weird situation in which case it's the graph's in like two parts because the x can't be zero. You can't have zero on the bottom of the fraction. So you end up with these two sections here. I'll try and make this as clear as I can. Um, so if I were going to draw this graph, minus 4, 6.5 is about there. 4, God. Okay, I think this graph kind of does that. I'm guessing slightly. Yes, it does. Um, yeah, because it can never touch that. Okay, and then once you've done that, the question says, use your graph to find the positive solution of that equals 0. Um, so it is y equals 0 is this line here. And so it's this answer here, wherever that crossed there. That would be the answer to that question. Um, okay, any other questions before we finish for today? Again, just a reminder for those of you in year 11 at least. Oh, there's more. Um, that is not an exponential graph. It is I don't know what the name is really. Oh God, what's it? Reciprocal graph, isn't it? Because when the x is on the bottom, that's what it is. Generally, they don't really look like that usually. It's usually a little bit more, it's just the scale's weird. But at the very least, that's what it kind of looks like usually. So that's the graph of 1 over x. So you have, it never touches either axis there and there. This one does because of the minus 2x bit, but that's what it is. Um, yes, sorry, finally, what I was going to say was the from the end of the month, I will be there, here every day, basically, for a while. How many of these graphs do we need to know? I'll tell you what I think you need to know. Roughly, you only get these occasional questions where it's like they give you some graphs and you have to say like which one's A, B, C, D, like they give you some equations. I would just say the following. Y equals x squared, a U shape. y equals minus x squared, a n shape or unhappy face, if you wish. Um, an x cubed graph can look, it sort of does that, essentially, an x cubed graph. Or it can be the other way if it's negative. Um, then, yeah, a 1 over x graph. So if the x is the bottom of a fraction, you get these sort of two pieces to it. Um, and was there one more? Yeah, I guess an exponential graph just means if it's like something to the power x, then it will always look like that. And you should also know that that value will always be 1, because when x is 0, what does that give you? Anything to the power 0 is 1. Can't think of anything else. I don't think you don't need to know those. That's it. I think that's probably it. Yeah. Although I don't see why there would be anything else that I can think of that you would need to worry about. But also, if you do get a question about a graph, I know this is very unlikely to happen in an exam, but you could make like one of those little table of values and just put some numbers in and find the y's and do a little sketch. And you can sort of figure out what it looks like, sort of, sometimes if you do that with a graph that you don't recognize. But it's a rarity for you to worry about. Right, assuming there's no other questions. But I will end it today. But I will just keep saying for you guys that you have a big advantage, which is just ask any questions, especially for the year 11. Contact us, use the WhatsApp, do whatever while you are revising to make sure you don't get stuck on anything. Um, so, firstly, on the further maths point, I mean, I don't know whether you are on TikTok, but in the last today, basically, I posted some videos, and there'll be more coming, which are all taken from further maths papers because some further maths questions are basically just very hard GCSE maths questions, some of them. And so what I did is I picked those out today, and I did quite a lot of those questions. However, I know you need help with further maths, and so I'm going to arrange some time for it. It might be during the half term, because I know your further maths exams are after the half term. Um, and there will be a bit more time and scope then. But in the meantime, if you have any specific further math questions, then just send them. I'll do them and it'll be fine. But have a look at those videos that I mentioned. If not, I can send them to you of just some random questions I picked out from a paper, which are really just slightly harder GCSE questions. Not even slightly harder, they're just like end of exam questions because that's what further math is. Right, that's all. I'll see you later.